This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hello everyone, and welcome back to Beyond Cancer. Today, as part of the 2024 Cancer Neuroscience Symposium series, me and my co-host, Dr. Nicole Sheff, will be interviewing Dr. William Hwang.
Dr. Hwang is an assistant professor of radiation oncology at Harvard Medical School. The Hwang Lab is focused on uncovering novel aspects of tumor-nerve interactome, driving disease progression, and they use single-cell and spatial biology, multiplexed imaging, and functional genetic screens in patient specimens and mouse models.
And
Dr. Hwang, we're really excited to have you here. Can you tell us a little bit about yourself and your research?
Sure. Thank you very much, Sahana, Dr. Sheff, for having me on this podcast. And for that kind introduction. So, as you said, I'm a physician scientist. I lead the laboratory of spatial and systems oncology, and my research background is actually in physics and engineering. So, I did my PhD in a pretty unrelated field in biophysics with Hwang at Harvard. And I was using and developing single molecule FRET tools to dissect how chromatin remodeling enzymes integrate histone acetylation signals and linker DNA length to create these evenly spaced nucleosome arrays that are a hallmark of transcriptionally repressed heterochromatin. So very kind of basic mechanistic work using kind of imaging, advanced microscopy, and biophysical tools. And when I returned to medical school after my PhD, I got really immersed in my clinical clerkships. And I just started to observe, what I gravitated most towards, because everything was intriguing to me. I could see myself in many different fields. And over time, I realized that some of my most meaningful experiences involved having the privilege of guiding patients through the terrifying experience of cancer. And I recall this vividly and really kind of etched my future path. I had this one elderly patient with pancreatic cancer, and he suffered from this horrible, intractable pain that we couldn't alleviate regardless of what we tried. The line between, him not being conscious and being in pain was basically, there was no line. So searching for answers, I went down to pathology, where I met an attending pathologist that I still work with to this day at Mass General to discuss the case as a junior medical student. And she was very kind. She took me through to the multi-head scope, and I peered into the microscope. She pointed out these extensive areas of this patient's tumor called that she termed perineural invasion, wherein the cancer cells essentially had invaded into the nerves. And the science hat that I had really was intrigued by this. What benefits, I was thinking, did cancer cells get from these nerves that they were invading, and vice versa? And perhaps taking a step further, how could we prevent this pathological collaboration from happening? So I filed this in the back of my mind because I had to continue my clinical training for several years. But when I returned to the laboratory, so this was in 2013, so this would have been 2018. So 5 years later, there was a lull actually during the pandemic where I actually got to think more broadly, and that's when I really got back into thinking about these cancer-nerve interactions. So I started doing a literature search, and I learned, surprisingly to me, that nerves within tumors outside of the central nervous system were not just implicated in neuropathic pain, which had been my initial observation, but also fueled tumor growth, adaptation to metabolic stress, immune remodeling, resistance to therapy, metastasis, you name it. And it became clear to me that if we could therapeutically intervene upon these tumor-nerve interactions, the potential impact on patient survival and quality of life was immense. But it was also clear to me as I reviewed the literature that we really needed a much better mechanistic understanding of how this cancer-nerve dialogue happens. And some of the literature that I saw, relating to central nervous system tumors at the time, was quite a bit more mature. But what I found, especially with pancreatic cancer, were hints, but nothing really that was comprehensive had spatial context, which I thought was really important for something like this, and also cell type specificity. So you knew kind of which cells were signaling with which other cells to create some of these emergent properties. So recognizing this, that, my small team at the time was developing and working with single cell resolution spatial biology tools, applying them to patient samples, stromal tumoroids, genetically engineered mouse models. It felt like we were well suited to contribute to trying to fill some of these gaps in knowledge and answer questions in this burgeoning cancer neuroscience field.
That's amazing. That's so cool and exciting to hear about. And going off of that, I saw that your lab has developed and optimized impressive single-cell and spatial bioinformatics strategies to begin to understand the perineural invasion in pancreatic cancer. Can you explain why the tumor nerve axis is so important in pancreatic cancer and what different cell type mediators your group has found that might be driving nerve recruitment?
Yeah, thank you for that question. I think pancreatic cancer, along with some other peripheral malignancies such as prostate cancer or certain head and neck cancers, for some reason have an incredibly high prevalence of perineural invasion. And the why question is one that's very intriguing, why it's so important. And we're really still in the early phases, I think, of uncovering this. So other groups have shown, for example, that sensory and sympathetic nerves, primarily through beta-adrenergic signaling, play an important role in pancreatic cancer development and progression. Even under certain conditions of metabolic stress, there was a really elegant study by Alec Kimmelman and colleagues at NYU a few years back that showed that if you deprive pancreatic cancer cells in culture or in vivo of serine, which some pancreatic cancer cell lines, serine is an essential amino acid, the pancreatic cancer cells can actually selectively increase their secretion of nerve growth factor and actually recruit nerves into the local microenvironment to then supply the cancer cells with serine. So, that type of collaboration, I think, 20, 30 years ago, certainly with peripheral tumors, was not something that we thought was happening. And I think everywhere we look, there's more and more studies now suggesting that this collaboration between pancreatic cancer cells and nerves is very important to the biology of this malignancy. So what my group did, a small kind of contribution that I thought we were well suited for was to undertake this comprehensive cell type specific and spatially resolved high molecular plex analysis of pancreatic cancer to start, but we're starting to move into other GI cancers. That's my clinical focus, so that's why we're going there first. In combination, as I mentioned earlier, with functional experiments using genetically tractable model systems. And what we're doing is trying to uncover the cancer intrinsic and extrinsic molecular mechanisms that underlie three very distinct processes. The first is nerve recruitment. So what are the cancer cells potentially secreting? What factors are they secreting that bring nerves to the local area? NGF, is one of them, as I mentioned, but there's many others that we're finding and in the process of validating. So I won't go into too many specific pathways today because I don't want to mislead the audience. There's still some preliminary results, but nerve recruitment is a big first step. The second, in the most histologically kind of apparent, is invasion, right? Perineural invasion. So the cancer cells in some cases will actually violate kind of the perineural sheath and enter, and that is what pathologists will pick up on and is very prognostic for patient outcomes. And then the third and related component is colonization. So once these cancer cells are inside this neural environment, how do they survive? How do they proliferate? What are they changing? We're noticing, similar to what people notice with brain metastases from, say, breast cancer or lung cancer, some of the metabolic changes there in their, for example, how they utilize different lipids is very intricately tuned to the local microenvironment. And some cancer cells cannot do that. And we think that's one of the reasons why certain aggressive cancer cell types, like pancreatic cancer, almost never see brain metastases, even though they invade the peripheral nervous system essentially 100% of the time. But yet we don't think it's an access issue. We think that perhaps it's an ability to colonize that microenvironment. Similarly, we're looking in the peripheral neural environment and trying to understand what are the mechanisms that allow a cancer cell to survive and proliferate or not in that peripheral neural environment. And I think you alluded to some of the computational work that we've been doing. So about a third of my lab is more on the computational side, but also to maximize our ability to predict dynamic cancer nerve interactions from static tissue slices. I've started a collaboration with Martin Hemberg's lab at Brigham and Women's. He runs like a computational single cell and increasingly spatial biology oriented lab. And we recently developed and applied this new method of inferring cell-cell interactions based on single-cell resolution spatial transcriptomic data that we call SCOTIA, SCOTIA, which is short for Spatially Constrained Optimal Transport Interaction Analysis. We recently posted that on bioRxives. It's in review right now, and for anyone who's interested, that code is available on Zenodo.
So I like the tie-in that you have, at least in your motivation to study these interactions from your clinical work to basic science. And so the pancreas is such a unique organ, given that it's innervated by a lot of different types of nerves, and you have this focus of trying to understand recruitment versus invasion versus why these cancer cell colonies can form in different types of tissue. Do you, from a clinical perspective, do you think understanding these three aspects of the tumor nerve axis will allow for earlier detection, especially in pancreatic, which really doesn't get diagnosed until late stage? Despite being densely innervated, it's able to grow undetected for quite a while. And so what is your goal in sort of trying to understand this tumor nerve axis? Do you hope for earlier detection, or do you hope to sort of harness this interaction for drug treatment?
Yeah, that's a great question, and I think we would all be in agreement with a cancer type like pancreatic cancer that any headway we can make on the early detection side is likely going to have much bigger, potentially exponential impact ultimately on humans than a similar kind of biological or therapeutic step in the metastatic setting, for example. So I think for sure that's something that we think about quite a bit. Another part of my lab, I won't go into this too much because it's less related to cancer neuroscience, but does look at liquid biomarkers, what are the earliest changes that correlate with in situ findings. We can do that most readily in mouse models, but we're starting to get the right patient samples to look at that as well. I do think the nerves are very much involved in kind of early cancer development, and therefore should give us a window into early detection efforts. But I also think that understanding those early interactions may allow us to modulate neural input and perhaps, for example, enable more effective anti-tumor immune responses, whether it's just the endogenous immune response or it's helped pharmacologically by us. On the first point, I think this work is well known now because it's maybe 7 years old, but Jamie Solomon, I think she was at Pitt when she did this work, showed in a autochthonous KPC model of pancreatic cancer that if you ablate sensory neurons, and she did this by neonatal capsaicin injection, I believe they found that you get significantly delayed formation of kind of precursor lesions called pancreatic intraepithelial neoplasias or PANINs, and that prolongs survival in these mice. This and other evidence that I won't go into really indicates that pancreatic innervation plays an important role, as I said, in some of the earliest stages of neoplasia. If we can find some of the specific pro-tumorigenic mechanisms that are enabled by these early cancer nerve interactions, I do think, Nicole, that we can likely find some new ways to detect and block early tumorigenesis without inducing significant degrees of neurological morbidity through non-specific blockade of physiologic neural signaling, which is always the worry, right? Is the therapeutic window. Because truthfully, we have a lot of therapies that can fully destroy a tumor. It's just what state do we leave our patients in after that treatment? So on the second point, yeah, exactly. On the second point, I mentioned, kind of, can we learn more about cancer nerve interactions to make anti-tumor immune responses more effective, which are quite poor overall in pancreatic cancer. Recent studies have revealed that tumor-draining lymph nodes are co-opted by tumor-derived factors. This leads to progressive immune suppression, including tumor-specific tolerance, resulting in distant metastatic spread. And while the immune and stromal populations in lymph nodes have been extensively characterized, much less is known about nerves that innervate the lymph nodes. And just down the road from me, Wiley von Adrian's group recently described two populations of nerves that innervate physiologic lymph nodes, including adrenergic nerves that regulate lymph node vasculature, and the sensory nerves that span the lymph node surface. And quite interestingly, I saw these movies from his group recently. Sympathetic innervation controls leukocyte motilities and immune response in the lymph nodes, including the ability to achieve antigen-specific killing of cancer cells. So when his group gave salmeterol, which is a beta agonist, you see all these moving CD8 and CD4 T cells in the lymph node essentially freeze in place, which is pretty interesting. The role of sensory neurons in training tumor-draining lymph nodes is not as well understood, but there is evidence that some of them activate dendritic cells to enhance pro-inflammatory cytokine production. And conversely, though, sensory neurons can also interact with cancer cells, promote T-cell exhaustion, reportedly through CGRP in these melanoma mouse models. So it seems like the crosstalk between nerves, cancer cells, and immune systems are complex and certainly altered in tumor-draining lymph nodes, in pancreatic cancer, and other malignancies. And if we understood kind of this three-way axis better, I think we'll be able to target these signaling pathways to reduce lymph node metastasis and improve therapeutic response, even at a very kind of early stage, to go back to your initial point.
Yeah, it's still going to require some ability, though, to pick it up earlier, I think.
Right.
Understanding more
Right.
how the nervous system changes, because I think from your perspective, treating patients, but from what we've heard, it's like patients experience some low back pain or some systemic shifts, and they don't attribute it to cancer necessarily, right? Once you talk to them, like, oh, you have stage 3. It's like, well, I did have all these problems, but they didn't think of it as a cancer issue.
Right.
Maybe if we have a better idea systemically of what shifts.
And going off of that, at least from like your MD, PhD perspective, what do you think are the biggest roadblocks to defining the nerve presence in the tumor microenvironment for better patient outcomes? And what problems are you hoping to see resolved in the future?
Yeah, I think that's a really nice segue, and I'll answer Dr. Sheff's question as well as part of that. I think you're completely right. One of the reasons pancreatic cancer is so deadly is the biological characteristics, the extremely desmoplastic nature, the kind of naturally built-in immune evasion, it seems, things like that. But it's also the late detection. I think that's totally right. The classic story is the grandmother that comes in because she's seeing her family for Thanksgiving, and they're like, "Grandma, why are you yellow?" And she's had this kind of painless jaundice and has been losing weight, but maybe it was trying to lose a little bit of weight, so it was actually happy for the first 3 months this was happening, but then lost 50 pounds and was like, well, I shouldn't be losing that much weight. So, there's stories like that. Once the pain comes in, that's usually, pretty progressed. I think we can catch the few patients that have chronic pancreatitis or who are lucky enough, in some ways lucky enough, to get an incidental finding. They have some gallstones and we do an abdominal scan and we're like, whoa, there's some sort of, lesion in the pancreas. Pannins, the most common precursor lesions, are not visible, but there's two other types, the mucinous cystic neoplasms, the MCNs, and then the IPMNs, the intrapancreatic mucinous neoplasms. Those are detectable by conventional cross-sectional imaging. So a lot of times we will risk assess those patients, take it out, because there's a risk of degenerating, and then we'll often notice, I'd say about half of the cases that I've seen, maybe a little less than that, there will be a small focus of invasive disease, and we're like, whoa, that was a great, a lucky catch, So outside of that, I think you're right. We need liquid biomarkers, ideally that, PSA is not perfect, of course. CA19-9 is far from perfect, definitely not a screening level test, maybe for tracking treatment in patients of the right blood type. But I think that if we understand the neural axis more, maybe there are some distinct biomarkers, hopefully kind of peripheral, but maybe even with, some advanced imaging techniques, again, the question is what's going to trigger, that imaging to begin with. It's going to, from a cost-effective standpoint, it's going to have to be patients that have a family history, right, or who have a known mutation, right, somebody with a BRCA2 mutation, or Leaf-Brown-Mayne, they will often be covered to get whole body MRIs, and then we can maybe pick up something. But yeah, a lot has to be done there. And then to go to Sahana's question, I think what intrigued me over the last, I've really only been thinking about this field for, heavily for the last 4 or 5 years. And it's interesting to me from a clinical standpoint that if you look at AJCC, right, which is what we use for staging cancers of all different types, the current edition features perineural invasion as a staging criteria for several cancers. You'll see it for several head and neck cancers, esophageal, thyroid, penile cancer, not pancreatic actually, and most patients have it. Perineural invasion is associated with worse clinical outcomes for many malignancies. We've shown this. We did a little pan-cancer study during the pandemic. Others have shown it for various cancers. And we actually already use the presence of perineural invasion to guide adjuvant therapy decisions. For me, kind of coming from the radiation oncology standpoint, we often add or intensify post-operative radiotherapy in the setting of perineural invasion for certain cancers. And one of the biggest roadblocks, I think, in defining nerve presence in the tumor microenvironment is our current binary classification approach. So I see this time and time again, right? If a tumor has a single focus of PNI and a careful pathologist sees it, it's annotated as PNI positive, perineural invasion positive, even if 99% of the cancer cells are nowhere near a nerve in the rest of that tumor. But we know this situation is almost certainly biologically distinct from a tumor with very dense innervation where everywhere you look, there are cancer nerve interactions, but we currently don't distinguish between these scenarios clinically at all. I think part of the reason why it doesn't seem important is because what are we going to do different about it, right? So I think understanding the nuances of these interactions and not just calling it one size fits all perineural invasion, but understanding, well, nerve subtypes, do they matter? They clearly seem to. Parasympathetic innervation of gastric cancer actually seems to be pro-tumorigenic and has the opposite effect of pancreatic cancer. So why, right? So we really need to understand this more, and then ultimately when we have more things we can do for different scenarios, I think we will hopefully start to see better stratification from a tumor nerve axis standpoint in the clinic. I will say one other thing about this. I think Nisha De Silva's lab recently had this really nice study. She used some similar tools to what my lab uses with spatial transcriptomics, and they showed that nerve proximity to cancer cells that don't meet perineural invasion criteria is actually prognostic. And just having cancers near nerves seems to induce those proximal nerves to have an injury pattern. So I think and they make a really nice case for this in their paper, I think it was published a year ago in Clinical Cancer Research, that going beyond this very kind of obvious perineural invasion, including proximity and other more histologically nuanced interactions between cancer cells and nerves in our clinical evaluation of tumors is important, but certainly has not been broadly implemented. So I think, that I think is another nuance that is a roadblock in translating some of our findings to the clinic. And perhaps finally, I'll just say that, as our molecular understanding of how the presence and activity of nerve-specific subtypes, as I said earlier, in the TME confers various benefits to the tumor, gets filled in more and more, we will hopefully have that clinically targetable node or nodes in cancer neuroscience to kind of usher in a so-called field of neurotherapy, kind of the way immune checkpoints with their discovery led to the explosive growth of immunotherapy. So I do think there's a lot of excitement and growing research efforts in the field. So I'm hopeful that over the next decade, we really will start to see these effective neurotherapies become a reality for our patients.
I really love that answer, and I love the focus on perineural invasion. It's something that I struggle with a lot when going through the clinical literature. And I love your research and your perspective because you have both the clinic and the basic science aspects as a physician scientist, and I hope that the listeners really do appreciate how we've interviewed a handful of people now and the differences between talking to a real basic scientist in the lab, just looking at animals, versus a physician scientist perspective of bringing in patient issues back to the bench and then vice versa. It's very unique and really special. And I really, really love hearing you talk about it. And we're really looking forward to your talk at the Cancer Neuroscience Symposium coming up in February. And we will wrap it up from here. Thank you so much, Dr. Hwang, for your time.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.